Thanks very much. Huntington's disease is one of the most common inherited neurodegenerative disorders, affecting around 1 in 7,500 individuals. Today, there are around 100,000 diagnosed patients and likely around 3 times as many carrying the causative mutation in the gene encoding Huntington. This mutation causes production of an aberrant form of the gene, which promotes transcriptional dysregulation, immune activation, and neuroinflammation. Earlier in the symposium, we heard Michael Hayden discuss the current status of disease understanding and the translation of that understanding to concepts for therapeutic intervention. We also heard from a panel of relatives, geneticists, and neurologists about the patient experience, prospects for improved strategies to address the unmet medical need, and how the clinical landscape is developing. In this session, we'll take a look at some of the therapeutic strategies currently being examined for Huntington's. We'll discuss how our understanding of disease pathophysiology is shaping our thinking on different treatment approaches and how these can be achieved. We will also consider how these strategies might be validated clinically. Our guests for this discussion are Ricardo Dolmetsch, President of Research and Development at uniQure, Irina Antonijevich, Chief Medical Officer at Triplet Therapeutics, and Amy Jackson, Chief Scientific Officer at Atalanta Therapeutics. I'd like to start with you, Ricardo. uniQure is currently evaluating the safety, tolerability, and efficacy of a microRNA gene therapy for Huntington's. Could you give us an update on the status of this study and tell us a little about the endpoints you're using to assess efficacy in the clinic?
Sure, thanks for the question. So we have dosed the first 10 patients in our study. It's a randomized placebo-controlled study. So this actually includes 6 individuals that got the therapy and 4 controls. This is the first cohort that got a lower dose of the gene therapy. Our gene therapy is administered surgically directly into the striatum. It is an AAV5 microRNA that is delivered to the region of the brain that is degenerating. And we will be assessing a number of endpoints. So we have a number of biomarker endpoints. These include mutant Huntington in the CSF, neurofilament light chain. We will also be using volumetric MRI to see whether we can halt the loss of volume in those regions of the brain. And then finally, of course, we will be using the United Huntington's Disease Rating Scale to look at the clinical outcomes. And then we have a number of exploratory endpoints, including some quantitative motor endpoints. So that's sort of where we are now. We will be enrolling the next 16 patients starting sometime in the second half of this year.
Perfect. Thanks a lot, Ricardo. So your strategy is to lower the synthesis of both mutant and normal Huntington. Is it fair to say that we're still at the stage of learning whether selective lowering of the mutant form of Huntington offers any advantage? Do the results of the recent Roche and Wave studies offer any insight into this point?
Yeah, so our strategy is to lower both the mutant and the wild type, and we believe that we can do this safely, and we think that this will be efficacious based on preclinical studies. So as you probably know, preclinically in an adult, it's possible to lower wild type Huntington almost completely, and it's still well tolerated. And of course, our preclinical studies in mice and mini pigs suggest that we can substantially reduce the expression of the mutant.
I see. Okay, thank you. Turning to you, Irina, at Triplet Therapeutics, you recently presented preclinical data for an antisense oligonucleotide therapy for Huntington's, targeting the DNA damage repair pathway upstream of Huntington. I wonder if you could help us understand the current status of translational models in this disease area and the degree to which such models are helpful in providing confidence in moving forward into your patient studies.
Yeah, sure. Thank you very much, Dave. So I would like to start by saying, I think the best model for our approach is really the human genetics. I think the last few years with the GWAS datasets have really firmly established the DDR, or the DNA damage response pathway, as a genetic modifier And what is particularly interesting, and I think that's different from mutant HTT, is that there is a clear gene dose effect. So we know from human genetic studies that individuals with lower expressing variants of some of those DDR genes show a greater effect on later disease onset and slower disease progression than those with a wild type. And also individuals who are homozygote for a lower expressing variant show the greatest benefit. So this is really human genetics that has really shed light onto the pathomechanism that is at play in HD. But of interest is also that there are several animal models. I mean, obviously there are many animal models that try to recapitulate HD, which of course is a complex disease. And so probably none of the animal models can do this fully, but multiple animal models exist that actually show somatic expansion and show an effect of a DDR gene. And in particular, we have presented the MSH3 gene as our target. And we know that in animal models, lowering MSH3 has, as we expect in humans, an effect on somatic expansion. And again, we can see this in a dose-dependent way. So we know that more lowering has a greater impact than less lowering. And on top of the animal models, we also have patient-derived cells that again show in vitro somatic expansion and an impact of, for instance, MSH3 lowering on the continued somatic expansion in vitro. So we think overall that the human genetics data, the animal data, and then the patient-derived cells really paint a strong picture in terms of the mechanism that contributes to HD. And what is, I think, also interesting in animal models is that both the human but the animals show somatic expansion, not everywhere, but in particular areas of the brain. And like in humans, these animal models show somatic expansion starting in the striatum and then ultimately also detectable in the cortex. And this is what we know from postmortem human data is also occurring in the human. So we used some of those animal models. We have actually used 2 different animal models, and in different animal models at different stages of the disease, to show that when we intervene and when we lower MSH3 in our case by about 50%, we actually hold somatic expansion at different stages of the disease. And maybe just to echo that this 50% is an important number, because we know from, again, heterozygote knockout animals that 50% really does have this impact on somatic expansion. We know from Vanessa Vila's data that there is an impact using CRISPR, and we have shown this therapeutically with our ASOs.
I see. Ricardo, you've recently published quite a lot of preclinical data as well. How do you put your recent data in the context of previous preclinical data, and perhaps how does it impact on your expectations from your clinical studies?
Yeah, we've spent, of course, many years trying to determine whether our gene therapy is going to be efficacious in patients. And so we've moved gradually from mouse models to most recently the mini pig model, which sounds like they're small pigs, but in fact they're full-size pigs. And so what we've shown recently is that our AAV gene therapy can reduce Huntington, mutant Huntington by 75% in the striatum, by 50% in cortex, that this results in substantial protection. And so I guess we're cautiously optimistic that this will translate into our clinical trial. I guess that's as much as we can really say. We, of course, fully understand that there's no preclinical model that is actually going to predict what happens in a very complex disease in humans. But we think that it's about as good as you can do preclinically.
I think in both of your cases, you're to a great degree reliant on the diffusion of your therapeutic entity through the brain to get to the right areas. And maybe, Amy, you can talk a little bit about your strategy with bifunctional siRNAs and perhaps whether that offers any advantage in brain penetration and diffusion.
Yeah, thank you, Dave. So the branched siRNA technology that we are leveraging at Atalanta was discovered in the lab of Anastasia Kurova at UMass Medical School. And it essentially consists of two double-stranded siRNAs joined by a chemical linker, each of the siRNAs being fully chemically modified on the 2' position of ribose at each nucleoside position within the molecule. And additionally, each siRNA also is asymmetric in nature, meaning that it has a single-stranded tail at one end of the molecule that is also heavily chemically modified. And what we're finding is that it's the complex interplay between all of these different aspects or components of the molecule that provides really fantastic both potency and distribution throughout the central nervous system. Some of the aspects of the molecule that we think can contribute to these very beneficial distribution properties include the molecular size of the molecule, which we think is related to prolonged residence time within the CSF. The highly chemically modified nature of the molecules has increased hydrophobicity, which enhances distribution and tissue uptake and retention. And then also the asymmetric nature of the tails, which are heavily phosphorothioate modified, enhances protein binding for biodistribution, and also the chemical modifications enhance residence time within the RISC complex. So what's been published by the Kurova lab in a groundbreaking paper in Nature Biotech in 2019 was showing really incredible sustained target knockdown throughout all regions of the brain, as well as the spinal cord after a single administration of the divalent siRNA. Now that we've brought this modality in-house at Atalanta, we have already continued to evolve and advance the platforms. We've already increased the potency as well as the duration of action. And we are also continuing to see this incredible distribution throughout the CNS, interestingly, in the absence of any conjugation strategy. Looking forward down the road, there's a potential to leverage conjugation strategies for cell-specific or region-specific distribution of the molecules. But at this point, we're leveraging the inherent biodistribution properties of these molecules for distribution throughout the central nervous system.
Got it. Thanks very much, Amy. Now, talking to all of you, there's a sense of increased disease understanding and unlocking of new therapeutic strategies, but also diversity in the modes that you're each using to use that therapeutic understanding and pathophysiology understanding. I'd like to discuss a little bit what you feel will be the next hurdles in further developing not only our disease understanding, but also driving that towards therapeutic benefit. Irina, perhaps you'd like to start.
Yeah, I think, thank you. I think what we all, are saying is that HD is a disease that affects multiple areas in the brain, and that brain distribution is really important. And I think we use different approaches towards that, but given that we don't know yet what will work clinically, I think it is good to have actually different approaches that are being explored initially preclinically and then of course also clinically. I mean, we know that it is a very complex disease, and I think having different approaches both from the modalities, from the target that we are targeting, but then also from the delivery methods, I think is actually great for patients, because we might also anticipate that not every patient, will respond to the same therapy. We know that there are variability between patients, and I think really the genetic modifier data have really exemplified that. And so I think it's an interesting time, an exciting time, and yes, there have been some setbacks, but I also think hopefully we will learn a lot from those setbacks to better understand what didn't work. Yeah, and I think maybe delivery is actually one aspect of those, So I think we are trying different approaches, and hopefully we will have data in a couple of years from now to be discussing, what worked and what maybe has to be improved.
Ricardo, would you like to pick up on any of these points?
Sure, absolutely. So let me add to what Irina just said. I mean, look, it takes a community to treat any disease. Huntington's is a devastating disease. And I think that the more shots we have on goal, the better it will be for everybody, right? We all want the same thing. There are many aspects of the disease we don't really understand. I mean, one of the things that we're struggling with now is trying to see which of the biomarkers are really predictive, and trying to assess what can be measured reproducibly, in these early stage trials. And I think we've learned a lot from the kind of groundbreaking work of Ionis and Roche. And I expect that the companies that come after us will learn from, whatever we're learning in terms of, how we measure, for example, the volumes. We would like to start using Huntington PET to actually see whether we can actually reduce the amount of Huntington or mutant Huntington in the brain. So all these things are going to just make it more viable. It's a journey. It's not the sort of thing that, any single individual or any single company solves.
Amy, that begs the obvious question. What do you think are the key learnings for you to take from the previous studies and from the comments from Irina and Ricardo as you're starting to plan your own preclinical optimization and clinical programs?
Yeah, I do want to reiterate what Irina said. I do think this is actually an exciting time in the field for Huntington's patients. Despite the recent challenges in the recent clinical trials, I do think we are learning a lot from those trials as to what worked and what didn't work. I think some of the key challenges that were approached related to aspects such as distribution and potency and tolerability. We now understand a little bit more about benchmarks as well as potential biomarkers that we can now implement early in our drug discovery and drug development processes to, I think, position us positively and favorably for being successful in subsequent clinical trials.
Fantastic. I'd like to turn a little bit to our broader understanding of neurodegenerative disorders and try and discuss a little bit how that understanding may impact on our thinking on treatment strategies for Huntington's. Do you expect that we may ultimately adopt some form of combination therapy approach, taking into account, particularly for patients that we're meeting for the first time in an advanced state of their disease, or at least a state of the disease with clinical symptoms? They're already experiencing some degree, at least, of neuroinflammation. Do you anticipate perhaps needing to not only treat regulation of Huntington levels, but also treat the neuroinflammation or immunomodulation aspects of the disease? Who would like to start?
I'm happy to start on that one. I think that's a really interesting idea and concept. I think clearly what we're learning is that Huntington's disease is a complex disease, and it's impacted by multiple cell types and multiple biological processes, and there's a lot of variability within the patient population as to the specific pathobiology that's impacted in their specific disease. So I think an approach in which we're taking a broader therapeutic approach to understanding each individual's biology and addressing multiple aspects of the pathology will be ultimately very beneficial to patients.
Yeah,
I think, go ahead, Ricardo.
Thank you. So I think whether one wants to use combinations or not probably depends on the stage of the disease. I think in some ways, in Huntington's, we're fortunate because the genetics are unequivocal. So we know for sure that expansion of the Huntington gene causes Huntington's. So in principle, if you can interfere with the expansion of the gene, either by knocking down the gene or preventing its further expansion early on, presumably that will treat the disease. I think what is not so clear is what happens once the disease has started. Because for many neurodegenerative diseases, it seems as if there's an initial trigger that then becomes a kind of self-sustaining kind of inflammatory disease. And so, for example, in Alzheimer's, people put a lot of stock behind trying to reduce A-beta. The genetics there were very strong as well. So obviously, if you produce a lot of A-beta, you will definitely get some kinds of early onset Alzheimer's. But it emerged that even if you reduce that early stimulus, you still get lots of neurodegeneration. So we're not quite sure what is going to happen in Huntington's. I think for all of us, the paradigm is really to intervene as early as possible. And if you're going to intervene later, then we might need, in fact, combinations that attack different pathways.
Yeah, I really very much concur with what I've just heard from both Amy and Ricardo. I really think that having a genetic diagnosis really allows us to intervene early. And this is where I think sporadic Alzheimer's disease has sort of challenges. So I think intervening early, and from what we know is that really the so-called pre-manifest, I think it's very important to stress that these individuals actually have symptoms. So it is not even a disease where we really start to intervene at an asymptomatic disease stage, but these individuals with pre-manifest disease clearly have biomarker changes. We know that neurofilament light starts to increase probably more than 20 years before the motor manifestation, but volumetric changes in the caudate nucleus and the putamen start to occur probably 10, 15 years before motor manifestation, full motor manifestation. In clinical science, the patients report cognitive deficits, behavioral abnormalities. So we know that there is something that has started. And so I think the earlier we can intervene to really hold this relentless progression and this neurodegeneration, I think the better. And I mean, of course, I don't know, but I think that the inflammatory signals that we see, even though when we look at PET data, we know that this also occurs, but it does sort of progress with disease progression. So I think it would be ideal to intervene early and maybe prevent at least much of, for instance, because you asked neuroinflammation, because we really hold the neurodegenerative processes early on. And we know that this can in itself be a trigger to, mount an inflammatory response.
I see, thanks. In some other areas, perhaps most notably in the ALS platform study design, we've seen the implementation of creative clinical development paradigms investigating the efficacy of multiple treatments in an integrated way. Do you think such paradigms have potential in the discovery of new treatment for Huntington's? Ricardo, would you like to say?
Absolutely. I think that there's a lot to be gained from, for example, sharing a placebo arm. The fewer patients that you need to not give a therapy to, the better off we are, and the better it is for the patient community. There's just a lot to be learned. There's a lot to be done also in terms of sort of synthetic control arms, where if we collect enough patients and look at their clinical progression, then you can subdivide them into cohorts that look like the patients that you're enrolling in your trial. So I think that can be very powerful. There are some practical issues associated with doing a genuine sort of platform, because all of these therapies need to be administered in different ways. So it's very difficult to keep them blinded. It would be difficult to do a trial the way they, for example, are doing some trials in ALS, or the way people are doing trials in Alzheimer's. If it's a pill, then I think you can blind it. If, on the other hand, you require intrathecal administration or intracisterna magna or ICV or surgery, that's going to be tough. But I think there's a lot to be done by sharing the control cohorts.
Right. Irina.
Yeah, I mean, definitely, I think sharing control cohorts, and this is where I think the Huntington's disease field has really resources that quite unique. I mean, we have a registry with Enroll HD where we have more than 20,000 individuals and data over, in some individuals, over many years. So I think that this is really a resource that we can utilize, and we can have, Ricardo's point, a much smaller, maybe still a control group within each trial, but then match it to the existing data, matching to the individuals that have actually been enrolled in the clinical trial. So I do think that this is important. In terms of triplets approach specifically, we are really very seriously planning for basket trials, because we have a molecule that it's not about multiple compounds in HD, but for us it is the same molecule in multiple indications, because it's a mechanism that holds in multiple other indications that we call repeat expansion disorders. And so we are contemplating to do a basket type trial where we can actually really also leverage particularly the safety and the dose escalation information from one trial for other indications, and then for each trial have, you know, a smaller data set, because you create a larger safety database across those different indications. And what is interesting in neurodegenerative diseases is also that you can actually even leverage some of the biomarkers, for instance, neurofilament light, but even imaging is applicable to multiple neurodegenerative diseases, which I think is also quite exciting. And then, of course, in our case, we also have our target engagement biomarker that we can utilize again across multiple indications that we think will really benefit, the patients across a whole spectrum of diseases.
Fantastic. Amy, would you like to add anything to that?
I concur with what I've heard. I think, you know, especially for a disease like Huntington's, where the patient need is so enormous and currently unmet, the better we can design more creative and adaptive clinical trials that actually can, you know, utilize smaller numbers of patients and get these therapeutics approved and to a larger number and broader population of patients, the better off everybody's going to be.
Thanks, everyone. Clinical studies today are necessarily focused on symptomatic patients. I wonder what hurdles you think we'll need to overcome before patients at an earlier stage of disease progression might become eligible for treatment once therapies become validated in the clinic. Do we still have a long way to go before we can envisage treating patients at a much earlier stage? Ricardo, would you like to discuss this point a little bit?
Sure. I think that there are essentially two things that we need to overcome. The first one is just the diagnosis issue. In Huntington specifically, but for many other genetic neurodegenerative diseases, people are reluctant to get diagnosed. There's a lot of stigma. There is just a lot of reluctance to know, especially if there is no treatment. So it's not trivial to ask people to do that when what you have is an experimental treatment. So that's sort of one part of it. I mean, the second part of it, of course, is that for people to be willing to enroll pre-symptomatically, or if not pre-symptomatically, at least as pre-manifest in terms of motor symptoms, you have to have a really safe therapy that is not a huge burden to administer. And you know, you're asking a lot of people. So it's, if the burden of administration is high, as it is for, you know, many therapies that we're talking about today, then I think it's going to be important to show at least some efficacy before people are willing to enroll really early. I think for some of the oral agents, it might be possible to do something pre-manifest, because I think the burden there is going to be lower. But even there, where there's potential side effects and tox, I think people are going to wonder whether they should enroll.
Yep. That makes good sense. Amy or Irina, would you add anything to that?
I mean, I would just want to add that I really agree that the benefit risk profile is very important when we consider earlier and earlier stages of the disease. In my experience working with the patient community, because again, the symptoms start early and they're actually impactful. Many patients cannot continue to work as they would like to. They have difficulties in their social life. So I think patients, particularly with HD, they know that motor manifestation is kind of like already many years down the road of their disease. So they're actually very keen to have a therapy that has a potential to really slow or even prevent further progression. But I think what is important is that nevertheless, it's not too onerous for them and that there is a good enough safety profile so that they are obviously not suffering from the side effects of the therapy. Yeah. So I do think that this is important. And I think what Ricardo said, the stigmatization, I think this is something that I think we all should work towards minimizing this, because I think hopefully we will one day have multiple therapeutic options for an early intervention. And I think we all think that early intervention has the greatest potential benefit for patients. And so we have to make sure that there is no stigmatization, that people have access to genetic testing and that they don't have any negative consequences.
Amy?
Yeah, agree. The cost benefit ratio has to be in the favor of the patient. I think, you know, we're all working hard to developing therapeutics that have improved safety profiles, also that are a lower burden for the patient. So that being either different routes of administration or enhancing, for example, potency or duration of action, so you can reduce the frequency of administration so that it becomes less of a burden, less of a challenge for the patients. And furthermore, we do have to demonstrate that these therapeutics are disease modifying. I think that's what's missing right now in terms of having molecules that are compelling for patients, particularly at early stages of disease, to be willing to take the perceived risks. If we could actually demonstrate now safe disease modifying approaches to Huntington's, it's going to be really game changing.
You reach at different stages of development and clinical validation of new strategies for Huntington's. I wonder if you could summarize in a few words your view on the outlook for therapeutic intervention for Huntington's over the coming years. Irina, would you like to comment?
Yes. Yes. So first of all, I do think that it is an incredible progress that we have seen over the last few years. I was in the HD field. I started maybe 11 years ago, and you know there was very little opportunity to even have hope. So I think we have multiple different opportunities now, and I think this is great. I think what is important is to have good clinical trial designs with good outcomes so that we can hopefully as early as possible in the development program and with as few patients as needed, but still enough to really have a conclusive decision, make a decision about what we said, efficacy, but also safety. I think working towards that is very important. At Triplet, we take this very seriously. We are developing additional assays to make sure that we can make this decision on both safety, signs for efficacy, as well as target engagement to select the right dose to actually be able to make these decisions.
Fantastic. Ricardo?
Yeah, I think that the future is as bright as it has ever been. It's of course been disappointing that the first set of ASO therapies failed. But at the same time, I think we have an understanding of the disease that we just never had before. And better than that, actually, we now have genetic therapies that just never existed. I mean, just the fact that we can safely deliver gene therapy into the brain that effectively would last you forever, that in itself is a bit of a miracle. And so in principle, we have ways of addressing these genetic diseases, that there are RNAs that have been modified so that they last a long time. That in itself is a miracle. That there are ASOs that can get to the right part of the brain is also, it's a huge advancement. I mean, from all the years I've been working on neurodegeneration, I think I see the the most potential for advancement now. So I think the future really is bright. And look, we're in a clinical trial. We will know sometime relatively soon whether this approach works, and we have the next generation coming along. We will not give up. I mean, none of us here.
Amy, Atalanta are coming into this field at an exciting time based on panelists' comments this afternoon. What are you looking forward to achieving over the coming years?
Yeah, I think it's a really exciting time in the field. I also agree. I think the outlook is very positive. We've learned a lot about this really challenging disease. We've learned a lot, even very recently, about different variations of Huntington itself that are implicated in the disease. We've learned a lot about orthogonal approaches to Huntington that can come to bear on the disease. We're learning a lot about technology development, chemical modification strategies, formulation strategies, device strategies to getting these molecules delivered to where they need to be and to have the right mechanism of action to be disease modifying. So I think for a company like Atalanta, sort of just stepping into the field, we're really fortunate to be standing upon the shoulders of some really incredible people who have done some groundbreaking work and positioned us, I think, very positively to hopefully really move the field forward.
Fantastic. Well, Ricardo, Irina, Amy, it's been a pleasure to talk with you today. Thanks for taking part in this discussion. On behalf of the Huntington's community, thanks for your continued efforts to find ways to improve the lives of patients. We all wish you every success, and I look forward to following the development of your programs. Thank you very much.
Thank you.
Thank you very much, Dave. Thank you.